Dear viewers, you are most welcome to my online class. Today we discuss a very important topic of plant physiology. It is glycolysis. Today we discuss what is glycolysis. Glycolysis is the common pathway of aerobic and anaerobic respiration. Here we explain it. 10 reactions of glycolysis. Easy way to remember 10 reactions of glycolysis. Calculation of energy production in glycolysis. What is glycolysis? Glyco means glucose and lysis mean breakdown. So glycolysis means breakdown of glucose. It is also known as Embden-Meyerhof pathway, in brief EM pathway. It is a process of formation of two molecules of pyruvate from one molecules of glucose by several oxidation reactions. It's a common pathway of aerobic and anaerobic respiration. Steps of aerobic respiration: first one is glycolysis, second formation of acetyl-CoA, third Krebs cycle, and fourth electron transport system or chain. Steps of aerobic and anaerobic respiration: first is glycolysis and second step is formation of lactic acid and ethanol. So we see here that glycolysis is the common pathway of aerobic and anaerobic respiration. This is first reaction of glycolysis. At first glucose is added with phosphate at 6 carbon position from ATP and converted to glucose 6-phosphate. ATP is converted to ADP by losing 1-phosphate. ATP means adenosine triphosphate. That means ATP has 3-phosphate. On the other hand, ADP means adenosine diphosphate. That means ADP has 2-phosphate. In this reaction, 1 ATP is used. Kinase enzyme is active here due to phosphate addition, and hexo word is used for glucose because it is hexose carbohydrate. This is second reaction. Here glucose 6-phosphate is converted to fructose 6-phosphate in presence of isomerase enzyme. Because these two compounds are isomer, glucose 6-phosphate and fructose 6-phosphate are isomer. Isomer has same components. The word phosphoglucose come from glucose 6-phosphate. Glucose 6-phosphate is substrate here and fructose 6-phosphate is product. In third reaction, here fructose 6-phosphate is added with a phosphate at carbon 1 position. From ATP, this phosphate comes from ATP and converted to fructose 1,6 bisphosphate. ATP is converted to ATP by losing 1-phosphate. Phosphofructo kinase enzyme is active here. In fourth reaction, fructose 1,6 bisphosphate is divided into 2 3 carbon molecules. One is dihydroxyacetone phosphate and another is glyceraldehyde 3-phosphate. In fifth reaction, dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by isomerase enzyme. These two compounds are isomer, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are isomer. As a result, two molecules of glyceraldehyde 3-phosphate are produced. In sixth reaction, two molecules of glyceraldehyde 3-phosphate are converted into two molecules 1,3-bisphosphoglycerate. Glyceraldehyde 3-phosphate dehydrogenase enzyme is active here due to removal of hydrogen from substrate glyceraldehyde 3-phosphate. Hydrogen ion is added with NAD, nicotinamide adenine dinucleotide, to produce NDS plus H plus. EI, inorganic phosphate, is added with one carbon position of 1,3-bisphosphoglycerate. In seventh reaction, two molecules of 1,3-bisphosphoglycerate are converted into two molecules of 3-phosphoglycerate by removing phosphate from one carbon position. 3-phosphoglycerate kinase enzyme is active here. Phosphate is added with ADP, 2-ADP, to produce 2-ATP. In eighth reaction, two molecules of 3-phosphoglycerate are converted into two molecules of 2-phosphoglycerate by transfer phosphate from 3 carbon, this is 3 carbon, to 2 carbon position in presence of phosphoglyceromutase enzyme. In ninth reaction, 2-phosphoglycerate is converted to phosphoenol pyruvate in presence of enolase enzyme. In 10th reaction, two molecules of phosphoenol pyruvate are converted into two molecules of pyruvate in presence of pyruvate kinase enzyme. Here 2 ADP are converted to 2 ATP. This is in reaction of glycolysis. Easy way to remember, if we know the product or substrate from each reaction, we easily remember these reactions. First substrate is glucose. Second substance is G6P, that means glucose 6-phosphate. Third product is F6P, fructose 6-phosphate. Fourth product is F1,6BP, fructose 1,6-bisphosphate. Fifth product is DHAP, dihydroxyacetone phosphate. Sixth product is G3P, glyceraldehyde 3-phosphate. Seventh product is 1,3-BPG, 1,3-bisphosphoglycerate. Eighth product is 3PG, 3-phosphoglycerate. Ninth product is 2PG, 2-phosphoglycerate. 10th product is PEP, phosphoenol pyruvate. 11th product is pyruvate. Glycolysis has 3 phases. These phases are, first one is energy investment phase or priming stage, 2 splitting phase, 3 energy generation phase. This is energy investment phase. First 3 reactions, 1, 2 and 3. Here 2 ATP are used in first reaction and third reactions. Fourth and fifth reactions are known as splitting phase. Fructose 1,6 bisphosphate is 6 carbon compound. It is divided into 3 carbon compound. One is glyceraldehyde 3-phosphate and another is dihydroxy acetone phosphate. 6 to 10th reactions are known as energy generation phase. In 6th reaction 2 NADH2, in 7th reactions 2 ATP, and in 10th reaction 2 ATP are produced. That's are all. Thanks to remain with me. Please subscribe my channel. Please like, comment and share. Allah Hafiz.